Thank you so much for joining us today and for giving us a little bit of insight into your project. Before we ask questions about the project, I'd love to know a little bit more about you and what your involvement is with HLH and histiocytosis in general.
The story really starts back in almost 2009. So when I did a postdoctoral visit to a lab in Switzerland, who was headed by Professor Jörg Chopp, and he was actually the scientist who discovered this inflammatory protein complex called the inflammasome. So as you can imagine, that name suggests it's highly inflammatory. And he characterized this protein complex that's inside histiocytes and showed that it mediates the production of these inflammatory mediators um, that have been known to cause inflammation in hemophagocytic lymphohistiocytosis. So the two cytokines he identified as being activated by this inflammasome complex were IL-1 beta and IL-18. And so during my time in his lab, I had the opportunity to study this inflammasome complex and do some molecular studies into how it's regulated. And so coming back to Melbourne at the Walter and Eliza Hall Institute in Australia, I managed to start my own lab here. And ever since that period in Switzerland, we've been looking at how these inflammatory cytokines are activated. And I think one of the standout findings in research in the HLH field in the past decade or so has been to show that in patients with HLH, they have very high levels of these inflammasome activated cytokines, IL-18 and IL-1 beta. And so that's where we started. We started asking, well, if there's a number of different types or flavors of inflammasomes, we started asking the questions about which type of inflammasome is activating these cytokines that are elevated in HLH, and also leading on from that, if we could model this in relevant animal models, what are the molecular pathways that lead to elevated levels of these cytokines? And so that's kind of where we started. And we ended up then looking at what would be, potentially novel therapeutic targets that we could then apply, sort of clinically relevant inhibitors to, which turned out to be these BET inhibitor proteins.
So, how long have you been working on this particular project?
Well, I haven't been doing many of the experiments. It was a PhD student. She spent literally 4 years of her life doing these studies. And a lot of them involved very late night experiments where we were. This model is obviously requires continuous monitoring in animals, and so we'd have to monitor these animals where we had knocked out what we thought at the time were the responsible inflammasome complexes, and then we had to monitor them over the time to see how sick they would get. And so she spent the last 4 years doing that, and we were very lucky to discover some of the key molecular players that are required for the activation of these inflammatory cytokines.
That was actually my next question. What are the key findings from this study?
So it's really interesting. There's the one major player that we think is going to be important moving forward is called it's a inflammasome protein complex called NLRP3. So that's the abbreviated name for it. And this complex, when it forms inside cells or it's activated, it actually ends up activating these enzymes in cells that cleave and activate the inflammatory cytokines. And so this is kind of the upstream target that you would probably want to inhibit if you want to block these inflammatory cytokines from being activated in patients. And I think moving forward, what's really exciting, there's a number of big pharma companies that have developed inhibitors of this inflammasome NLRP3, and there's actually quite a few ongoing clinical trials, particularly in Parkinson's disease, using these NLRP3 specific inhibitors. But I think there's a good chance moving forward that they could also be applied to HLH. I guess what's nice to know, we're not the first to implicate NLRP3 in HLH-induced activation of these cytokines. There's been other studies sort of suggesting NLRP might play a role in different HLH animal models. So I think it's a very exciting target to pursue.
That's fascinating. So, what do you think are the future potential implications of this study for the patient community?
It's early days, so we only used one HLH model, I'd say. So we identified a number of key molecular players. So we identified NLRP3. Then we also identified a couple of other, these are pore forming proteins downstream of NLRP3 that allow the release of these cytokines. And these pore forming proteins are called gasdermins. So apart from identifying a number of potential therapeutic targets, I think the next steps would be to examine the requirement of these key players in different HLH models, and then ideally manage to get some human cells from patients with HLH, and then we could test properly if the same targets are responsible for the activation of these cytokines in the human cells. And then from there, I think there would be a very strong rationale to use the clinically trialed NLRP3 inhibitors in HLH.
That's great. It sounds like this could really help this whole inflammasome process for HLH patients, which is really exciting, I think, for them to hear. Thank you so much for taking your time and for the hard work that you're doing. Our community loves to hear about all of the dedication for the physicians and the researchers, and it really gives them hope moving forward in their own journey. So, thank you so much for your time.